Hello, my name is Karina Cabrita and I'm a product manager at Cytognos. It is my pleasure to introduce to you our invited speaker, Elaine Sobral da Costa, which will present to you the clinical value of advanced software tools for the routine workflow in a pediatric center. Elaine is currently a pediatric hematologist and head of the flow cytometry department at the Federal University of Rio de Janeiro in Brazil. Elaine had her degree in medicine from the Federal University of Rio de Janeiro in 1996 and her specialization in pediatric hematology from the same university in 2000. In 2004, she started a split-site doctoral program between the Federal University of Rio de Janeiro and the University of Salamanca. Ever since, she has been a frequent collaborator with the University of Salamanca in the development and validation of new panels. Elaine is the inventor in several international patents and has her publications in several reference journals such as Leukemia, Blood, PLOS ONE, and Cytometry A. Also, she is currently an active member of the Euroflow Consortium. Welcome, Elaine, and thank you for being here today.
Hello, everybody. First, I would like to thank you, Karina, for the kind words, and also thank you, Cytognos, for the invitation. Today, we'll talk about clinical value of several software tools to flow cytometry in a routine workflow in a pediatric cancer. As Karina said, I worked in a pediatric institute from my university in Rio de Janeiro. And we mostly work there with acute leukemias, also MRD in acute leukemias, primary immunodeficiencies, and also solid tumors. So several fields in biology use large data set of information, as for instance, microarray, metabolomics, proteomics, next generation sequence. But nowadays, flow cytometry has becoming a big data set area because the number of parameters studied by cell, and the number of cells studied, they together perform a matrix by far larger than matrix from microarrays, for instance. So, despite this big advance in day by day on flow cytometry data analysis, most of service used the Boolean strategy, that is, we perform the gate, for instance, here, and we see the cells in other parameters. In that time, we had a few parameters per tube. So we put, for instance, CD45 in several tubes to see how these cells, CD45 low, were in different aliquots from the same sample. And after we joined in our mind all this information to say what these cells are. So before to comment these applications, we have to start with standardization procedures, because it's impossible to compare data that are not standardized. A little bit of history. It's true in Brazil, but I think in many countries it's the same thing. Many flow cytometrists started with basic research and after they went to work with clinical applications. So, in basic research, they had every time a control in the same time, the same experiment of all these experimental conditions. They have the same genetic background, same lineage. We, in clinical flow cytometry, when we have a normal control, not necessarily this guy have the same genetic background of our patient. When I have our patient in different conditions, it not occurs at the same time. We have, for instance, a patient with acute leukemia. 15 days after, we have to evaluate him to find his cells, his leukemic cells, 33 days, 7 to 8 days. So it's not possible to have here, for instance, all necessary controls as a normal control, cells from the diagnosis. So what we have to do is standardize the calibration, compensation, and all the whole procedures to make the cytometer comparable during the time. So in basic research, many guys to adjust the voltage for each experiment. But for the clinical research, for the clinical applications, it's not possible. We have decided to use it. The Euroflow group have standardized the use of beads of calibration, that's rainbow beads, and we use the 7th peak of fluorescence, and we have target values, and we can accept a small deviation, and we have to do the calibration after we use the automatic compensation of the cytometer, but with all tubes, all controls, 1 tube per tandem. Everything is described in SOPs of Euroflow site. And after, we have to verify daily if the calibration is okay, and we use it daily, this rainbow bead, and we accept a lower deviation of 1% of the target value if the parameter are systematically deviating day by day. After 3 days, we have to recalibrate the equipment. So we are able to maintain our machine comparable during the time. And also, we have now several machines in different labs completely comparable. It's nice because we get to have many samples from different countries to establish a database for several diseases in Euroflow consortium, but also in day by day, it's very interesting. In my center, we don't have a bone marrow transplantation for children. So when one patient needs to a bone marrow transplantation, he sent to another center. If the center use the same strategies than our strategies, if the center follows, for instance, Euroflow procedures, we can compare our files, previous the bone marrow transplantation and the files after, and it occurs nowadays. We worked, for instance, with a center in Jaú, in São Paulo, and they use the same SOPs and the same panel for MRD detection, for instance, and we can compare the MRD before and after the bone marrow transplantation in different centers, and the data is fully comparable. Here we have the example of the quality assessment of Euroflow that compares samples from different labs in different countries, and they give us our scores. Here we have all of parameters inside the good range, and only one parameter in one sample that was outside of range. So we had a very good result, and we can also learn more and more with this process. Before to beginning, we have also to compare basic research to clinical flow cytometry concern controls. In basic research, we have to have 3 repetitions or 5 repetitions of one experiment, for instance. In clinical flow cytometry, we have a huge variability in the controls, so we have to have higher numbers of controls paired by age, by gender. It's an example of work from Euroflow in primary immunodeficiencies. So they have studied 200 of health donors, split by age range, to build the normal controls for this study. So now we have to enter in our objectives for this speech. So first, differential diagnosis in acute leukemia. Flow cytometry is used since the 80s for this purpose. In that time, they had morphology and they had flow cytometry with Boolean strategy and few parameters. And now what we have, they have to use it only the accumulate knowledge about hematopoiesis, about cell differentiation, normal cell differentiation patterns, and also the experience accumulate with the day by day and with the seeing case, different case, to identify, for instance, that this case is a T-cell acute lymphoblastic leukemia, because expression of CD7, partial expression of CD3, cytoplasmic CD3, and so on. So what we think? If we have a database of leukemic specimens, why not calculate the probability of new case belonging to a certain pathological category, for instance, T or BCP, acute lymphoblastic leukemia, or acute myeloid leukemia, or mixed phenotype. So The first problem is that it's difficult to calculate probability distribution in a high number of dimensions. So the first solution we have used was a technique named principal component analysis that is hugely used in other biological problems, such as microarrays studies. So principal component analysis is a linear combination of parameters. So we attribute, it's totally non-supervised. So the methods attribute height for each parameters, and these heights are adjusted to achieve the maximum variance among the events. So we have here the populations split in one graph, two-dimensional. Further, we can discover these weights if we click with right. The diagram info have these weights. For instance, here CD3 was multiplied by 29, CD3, cytoplasmic CD3 was multiplied by 23. So these weights are from this first principal component, but we can have also the parameters for the second or the third and so on. Next, we can put together files from different patients with the blasts identified. So we can have the probability distribution of each population with the median probability and all the whole distribution. And we can see it in a different way, like this way, when each dot is the median point of each case. And we have two lines here, one line corresponding to the first standard deviation and the other line corresponding to the second standard deviation. So how we can use it in the real life? Here we have a 5-year-old boy with a suspicion of acute leukemia. We received his bone marrow, and we have here, and it's an easy case with blast CD45 low, with expression of CD19 and cytoplasmic CD79A that are classic B-cell markers. In absence of CD3, CD7, and intracellular CD3, but with expression of CD34, showing that they are immature cells, but this is the case had a small part that have a low expression of MPO. So we can put this case in the database. We click here in load database. We have to inform the age of the boy, 5 years old, the sample, if it's a peripheral blood, lymph node, bone marrow, so it's a bone marrow, and we have informed each population we want to compare with the database. So we have here our tube, our acute leukemia orientation tube with the markers I have showed in the previous slide. So we choose principal component analysis separation, and the program inform us this compass pointing 79% of probability to have a B cell lineage, and also 19% of probability of a mixed leukemia. And further, we can see my patient here when compared between a database of cases of T-ALL cases compared to BCP ALL cases, and my case fits here. Also, when compared T with myeloid leukemias, my patient fits in myeloid leukemias. When I compared B with myeloid, they fit in B, but some events are outside the B cell database. And when we compare our case with B case only, we have part of the population inside the B cells and part outside. And when we compared our case with myeloid case, it's completely outside. So we have expanded our panel to investigate this case, and in the end, he was a boy with BCP acute lymphoblastic leukemia, but with a low expression of MPO without any expression of any other myeloid marker. Here we have also another information that is a robust comparative, robust curve. This comparative plots every case we have in the database in order, and we have our case compared with these cases, and our case is outside from T, outside from myeloid, and our case fits inside one BCP case. The median of our case is identical of another case from our database belonging to BCP ALL. So this information is quite useful in our day-by-day. Further, we are in a university hospital, so we have many students there, and we can use this tool to our students for they check their knowledge. So they analyze manually, and after they have to compare with this compass analysis to check their answers. So it's quite nice for learning also. This tool, it takes few seconds speed to run. Next application is the evaluation of minimal residual disease in B-cell precursors acute lymphoblastic leukemia. So we used the same strategy. For instance, the calculation of the probability distribution of each population, normal versus leukemic. Instead, several classes of disease. Here we have disease against normal, but our normal in BCP, in B-cell precursors, it's a pathway. It's not a cell population isolated. So here we have used a strategy to select our B-cell precursors, runs a principal component analysis. So just to share more immature precursors, CD34 positive, and the precursors CD34 negative, the normal precursors. After we can fix this transformation without changes, and we can compare our case against these two populations. If my case is similar to more immature ones, I can take out the other B precursors and runs again a principal component analysis, comparing my blasts with normal cell precursors. So we can have, for instance, a situation like this. That's an ideal situation. It's completely splitted. The two populations are completely different. And we can have a worst case that the two populations are superposed in the two standard deviations. So in this work from Prisca, from the whole Euroflow, we have used this strategy to build the BCP ALL panel for MRD detection that in 2017 that we have used in the lab and many centers around the world are using. And this panel had several rounds. Each round was evaluated with this strategy until all cases can be identified with the final panel. But we have talked about the maturation as a continuous pathway. So it's occur under MRD condition in all acute leukemias. So we have also studied the principal component analysis to reduce dimensions in pathways. For instance, PCP maturation, but it could be, for instance, for myeloid populations as neutrophil differentiation or so on. But now the example is in BCP. So we have a started point and in 34 positive cells, and we have here arrow defining the root of this pathway, and this pathway in several normal samples can be normalized as number of standard deviation during the pathway, and we can also compare the new case. For instance, we have here B cells. We can select these B cells with CD19 and compare it directly with this pathway. Now we can see that in this stage we have some markers outside the two standard deviation pathway. So we have checked the maturation seems quite normal. We have here cells CD30, CD45 low, small cells that mature, gaining CD45, starting here, loss in CD34, and now after CD38, and also here with CD10, CD20, the normal pathway, but here we have some events, 79 events outside the maturation pathway. So this tool can be also useful for us. But we have much more now. We had collected normal case and reactive case for several samples to build a normal phenotype library, putting together reference files from different ages and so on. So we have to inform age of the patient and material and so on. And after the program to start with two clustering events. So for each event, for each cell, they look for the nearest cell in all parameters. This method is named nearest neighbor. It's the same method used in other software tools, in other software, including outside from Infinicit, for instance, SON, and in Infinicit for calculating data, but this is used in automatic gating to first clusterizing the events, and next each cluster identified is compared with cell population that was described in our normal library. So if the cluster is completely matched with our reference population, it's labeled as such population. But if the cluster is outside all populations, or if they are partially in a population, cluster is put in check population. So we'll see we have several options to identify our cluster. So it's again, we have a real life case from our lab. It's a girl, 3 years old. We have her bone marrow, and with 15 days after the induction, and we have to also inform the panel. We have to have the 2 tubes for use this tool, but normally it's not a problem in our lab. We routinely use these 2 tubes. We not choose one or another tube according to the phenotype. So this tool takes some minutes, 7 to 9 minutes more or less. We can take a coffee and so on. When it runs. When it finish, we have to check some populations. Normally it's a few percentage of events to be checked. If everything is okay in the standardized procedures and with the machine and so on. So it's also can be used as a kind of quality control in day by day, because if we run the automatic gate and systematically we have more than X% in check populations, if we have many events to be checked, we have to think about what is wrong in our procedures. Let's see, for instance, our check populations. We have here, for instance, preview 1 cells to be checked. They suppose that preview 1 cells, and they put all these options. These are very small cells, but the phenotype is CD10, CD20 positive, CD34 positive with CD38 negative, with the expression of CD66C plus CD123. Most of these events are in the first 2, so the events in the second tube were better identified for some reason. So we put these events inside the abnormal precursors. But first, we also plot all the other B cells to be sure. So we have here, for instance, the normal B cells and the plasma cells to be sure. Also, I decided to show it. We have in this tree of populations, we have also mesenchymal and endothelial cells that in many labs they are not identified in the analysis because of the time, because of several reasons. So we have many populations identified here in the normal library. So the program correct identified for me, they put in the check, but CD45 negative, large cells with CD34 expression, they are not doublets, they don't have CD20, CD10, CD38, and they have a high expression of CD81 and CD73 is expected for endothelial cells. So we can check as endothelial cells. So we click here and the program put these cells here. And the final analysis, we have our blood cells identified here. The percentage was quite similar of our manual analysis, 0.02, but we have also what are the parameters that are outside the 2 standard deviation for our cell population. In other words, we can inform exactly what parameters were important to define MRD in these cells and in the report. Also, we have automatic gating for primary immunodeficiencies. As you see in the first part of the presentation, they have accumulated a huge number of normal cases to build this database, normal database. So we have selected here again the sample, in this case a peripheral sample, and PDOT, and we have to write here the age that I forgot to put, and we have to select the automatic gating and identification. So again, we have to wait a little bit, around 10 minutes more or less, for my computer. And I think this screen is quite interesting because we have here, we have to also inform the total leukocytes or total lymphocytes here to calculate the absolute number of cells. And here we have interesting pictures. I have put them bigger to see. And we have regions of normal cells. And we can see here, for instance, in the CD8 cell populations, we have 2 cell populations empty. In fact, the other goods about the automatic gating in primary immunodeficiencies is the automatically comparison with reference values for each cell population. So we have here absent populations, CD8 positive, effector terminal differentiation, CD27 gene, and 27 negative, and also B cells, IgD positive, IgM negative, post-GC. These cells are absent. And finally, the program suggests us comments. With this range of reference, it puts which population are in the normal reference range and what is not in the reference range, what is decreased, what is increased, and so on. So its tools have helped us included in timing to write the reports. We have to check everything, but it takes less time than to identify all populations and to type in every number. So it's quite useful. Finally, we have talked about building our own database. For instance, for designing validated new panels. I have the solid tumors example. We are studying pediatric solid tumors since 2009. We have published this paper in 2013. And now, in that time, we had 52 samples, and now we have more than 400 samples of pediatric solid tumors. And another paper is coming. So in that time, we have worked in 2 steps, sequential steps. So first step, we have used some strategies to identify tumor cells. So the most useful markers in that time were CD45 and CD56 to identify cells with high expression of CD56 without expression of CD45, and they are completely different for monocytes, neutrophils, eosinophils from all hematopoietic cells, also from lymphoid cells. So we could properly identify neoplastic cells, but we could not classify from each tumor or these cells. So we have used a huge panel of characterization. In that time, we didn't have some markers conjugated. It was improving along of time, and now we have more markers studied and more markers conjugated. It's a little bit bigger than I show here, and we have done for each tumor cell population for each patient. We have done one file, merged and calculated, with a total of 45 markers. So we have put together all antibody combinations from a same patient, from a same sample, and we identified tumor cells as CD56 positive, CD45 negative cells in each antibody combination. Further, we have calculated using nearest neighbor technique that can calculate one event in 1, 2, how should be this event in the other 2 if it was stained with the other antibody combination. So at the end, we have all parameters in all events for all events of our tumor cells in one sample. So we can put together as an initial in-house database cells from several cases of neuroblastoma, of extraosseous Ewing sarcoma, from rhabdomyosarcoma, and we can initially compare these populations. For instance, PNET is the same of extraosseous Ewing sarcoma compared to rhabdomyosarcoma. For instance, the cases in that time were few cases, but we could identify markers with high weight in this separation. For instance, CD271 and GD2. Comparing neuroblastoma with Wilms tumor, we have identified GD2 that's high expressed in neuroblastoma. It's not expressed in Wilms tumor. And EpiCan that's expressed in Wilms and not expressed in neuroblastoma. And also we can compare neuroblastoma and extraosseous Ewing sarcoma, and we have found CD99 that's expressed in PNET and not expressed in neuroblastoma, and GD2 that's expressed in neuroblastoma and not expressed in PNET. CD271 is super high in rhabdomyosarcoma. Is expressed in PNET, not so high as in rhabdomyosarcoma, but it's a strong positive. And GD2 is low expressed in PNET, and it's not expressed in rhabdomyosarcoma, for instance. In that time, we had a few cases in some categories. Now we have much more, and it will be better. this selection of markers will be better. But this in-house database was quite useful to identify markers, further test in a propose of solid tumors orientation too. So we can build our own database and cases of new proposals as pediatric solid tumors diagnosis, or also in case of we don't have some marker in our panel, we had done some deviation of the panel and we have to build our own database to have our normal controls and so on. We can easily do it. So I will wait that the examples of our day-by-day routine were useful for you. I have to acknowledge very, very, very much to the Euroflow group. I participate of the Euroflow since the beginning, and I have learned very much from everyone there, and also a huge work was done by the group to achieve all these tools and all these results we have today to facilitate the cytometry's lives. And I have to acknowledge my team on our lab, and especially Alberto Fal, Carlos Eduardo Pedreira, and also Quentin. Where is Quentin? Here. They were very, very important in the construction of all these tools for our use. Thank you very much.